Hello. Welcome to the next in the series of review clips designed to assist you in preparing for your Biology 3201 public examination. In this clip, we'll examine the concept topic of mutations. We define a mutation as being a permanent change in the genetic material of an organism. All mutations are heritable. That is, they will be copied when DNA replicates. Yet, not all mutations are passed on to the next generation. Only germ cell mutations in sex cells, will be passed on to future generations. Somatic cell mutations, that being mutations that occur in body cells, will not be passed on to offspring. Rather, they are passed on to new cells created in the offspring in which it occurs. Germ cells, remember, create sperm, create eggs. When sperm and egg are mutated, this in turn results in the offspring receiving the mutation. Yet in a body cell mutation, when it occurs, only the new cells in the body will receive that mutation. Mutations fit into one of two broad categories. We have gene mutations and we have chromosomal mutations. Gene mutations occur when one or a few nucleotides are changed within a gene. These are often referred to as point mutations. There are two basic types. The first of these is substitution. A substitution mutation is when one nucleotide is replaced for another. For example, CAT becomes CAT TAT. The second C was substituted by a T. Or the fat cat ate the wee rat became the fat hat ate the wee rat. Again, a simple substitution for an H for a C. Substitution mutations may or may not have an effect on cells. It depends on where and what nucleotide is substituted. There are three possible types of substitution mutations. The first of these is silent. A silent mutation is a substitution that has no impact on the cell's metabolism. We have a missense mutation, and a missense mutation is a substitution that creates a slightly altered, yet still functional protein. Granted, these mutations can be harmful. In fact, a single nucleotide mutation in the protein that makes up hemoglobin results in a disease known as sickle cell anemia. And we also have our nonsense mutations. A nonsense mutation is one that renders a gene unable to code for any functional protein. It usually occurs when the start signal is erased or the stop signal is prematurely inserted. Nonsense mutations are always harmful. The second type of mutation, gene mutation, are those which return to be frame shift. Frame shift mutations result from the insertion or deletion of one or two nucleotides within a sequence of codons. This caused the entire reading frame of the gene to be altered. It caused the sequence of amino acids of protein to be changed from the point of mutation forward. Frame shift mutations fit into two broad categories. The first of these is insertion. An insertion occurs when an extra nucleotide is inserted into the DNA sequence. This would push the reading frame forward. We also have deletion. And a deletion occurs when a nucleotide is actually removed from the DNA sequence. The example you see illustrated in sentence form is as follows. As you can see, not only is the original codon affected, but every codon from that point forward is also altered. The other type of broad type mutations are those we deem to be chromosomal. Chromosomal mutations affect entire chromosomes, parts of chromosomes, or even multiple genes on the same or different chromosomes. These mutations lead to the rearrangement of the genetic material in the chromosomes themselves. There are a number of different types of chromosomal mutations. The first of these is transposons, or jumping genes. These were first discovered by Barbara McClintock, and these are genes that are able to move or jump from one area in the DNA to another. This leads to tremendous variety in organisms. Indian corn has a variety of colors as a result of transposons. Deletion, another type, is when a portion of the chromosome is lost. Part of the chromosome often breaks off and is lost entirely. This could be a result of viruses, radiation, or chemicals. One example is a deletion on chromosome 5. When a piece is lost off of chromosome 5, children are born mentally handicapped and with a cat-like voice. This disorder syndrome is called Credit-Shat syndrome. You can see this illustrated in the diagram. In here, the original chromosome was A, B, C, D, E, F, G, H, I. When mutated, the section D through G become deleted, resulting in a chromosome of A, B, C, H, I. Another type is inversion. In inversion, a section of the chromosome is exactly that, inverted. Here, a segment of the chromosome may break free and reverse. It then becomes reconnected. Autism. Some people believe this might be one of the reasons for autism. As you can see in the diagram, what happens is the original sequence, A through I, was altered. D, E, F became inverted, such that it now reads A, B, C, F, E, D, G, H, I. This would be an inversion. Duplication is another type of chromosomal mutation. With a duplication, a gene sequence may duplicate itself one or more times within one or even several chromosomes. Repeats are not always harmful, but they can sometimes affect the functioning of a gene. Fragile X syndrome is one example of a disorder caused by that. Here, the X chromosome in some females and in males have several repeating units of nucleotides. Again, you can see illustrated. The original sequence is A through I. With sections C and D removed, duplicated, and reinserted, it now reads A, B, C, D, C, D, E, F, G, H, I. The C and D section were duplicated. Translocation is another type of chromosomal mutation. Here, a part of one chromosome changes place with another part of the same chromosome or with part of another, a non-homologous chromosome. It often results in one longer and one shorter chromosome. Some types of cancer are caused when chromosome 14 exchanges place with section on chromosome 8. In fact, some of the symptoms attributed to Down syndrome are related to translocation between 14 and 21. And you can see this illustrated in the diagram. We had two separate chromosomes, A through I, J through R. J and K were removed from the second chromosome and reattached to the first, as you see illustrated here. This represents a translocation. Non-disjunction is another type of chromosomal mutation. Here, we have a failure of a homologous chromosome to separate during meiosis. The result is a cell with either too many or even too few chromosomes. Of the cells created, one will have an extra chromosome, while the other will have a missing chromosome. These fit into two main types. Trisomy is when an extra chromosome is inherited in a cell, resulting in 3 chromosomes of the same number in a cell. Trisomy 21 causes Down syndrome. And we also have monosomy, and here only one chromosome is inherited instead of a pair in a cell. Public examinations have asked questions on this topic, and I present the following. In which situation could a mutation be passed on to the offspring of an organism? Your choice here: a cell in the uterine wall of a human female undergoes a chromosomal alteration. This would be a somatic mutation, which would not be passed on to offspring. The DNA of a human lung cell, choice C, is mutated. Again, this is somatic. And certainly D is also representing a somatic mutation. Remember, only germ mutations, those that affect egg and sperm, are passed on to the next generation. Therefore, the answer is indeed B, if primary spermatocyte in a human forms a gamete containing 24 chromosomes. This question asks you what type of mutation occurs if the DNA sequence CCG TTT CGG changes to CCG TTC CGG. The change occurred in the third codon, or the second codon, third letter. The T was replaced by a C, or what we had was a substitution. And finally, you're asked which represents the situation below. In mutation 1, the normal DNA strand was AGT, CGT, AAG. The normal had an inversion in the second codon. Only the second codon was affected, however. 1 and 3 remained as was in the proper sequence. In mutation 2, ABC became CBA. Here, all three mutations, all three codons were changed. As a result, the more severe mutation would be number 2 and has a higher number of genes affected. Thank you for your patience. I hope you found this learning clip to be helpful.